Hello students, in today's class we are going to learn about different environmental factors that affect the growth of microorganisms. So here are the list of some of the environmental factors that influence microbial growth, which include temperature, pH, osmolarity or water activity, oxygen level and UV radiations. So microorganisms try to adapt to the variations in these different environmental factors through one or other mechanisms. Some of the organisms are able to adapt to the extreme environmental conditions. These are known as extremophils. So we will be looking at what and how each of these factors affect the organism. So coming to the first factor, that is temperature. So at a very lower temperature, the membrane is in a gel-like form and all the cellular processes are also slow. As a result, the growth is slow. As the temperature increases, the enzymatic reactions that take place at a starts to take place at a higher or an increasing rate, and until we reach a temperature which is known as an optimal temperature where maximum enzymatic activities are possible, and hence growth rate is also maximum at the optimum temperature. From here, as the temperature further increases, the growth further decreases because with further increase in the temperature, it can lead to protein denaturation, collapse of the cytoplasmic membrane, and ultimately thermal lysis. However, not all organisms have an optimum at a temperature of around 30 or 37 degrees Celsius, but microorganisms have a different or a varied temperature optimum. Like we have psychrophills which grow at a temperature range of about 0 to 15 degrees Celsius. We have mesophills that prefer 20 to 45 degrees Celsius. Many of these are pathogenic organisms that grow in living organisms such as humans around temperature of 37 degrees Celsius. So then there are thermophils which like higher temperatures about 45 or above 55 degrees Celsius. And then there are hyperthermophils which can grow at very high temperature of about 95 plus degrees Celsius. So the enzymes that are used for PCR obtained from Thermus aquaticus is one of an example of a hyperthermophile. Then coming to the next factor, that is pH. Based on the pH requirement or liking towards the pH of organisms, they can be categorized into three types. Neutrophils that will grow in a neutral range between slightly acidic to a slightly alkaline range between 5.5 to 8 pH, while acidophils are those which like acidic range ranging from 0 to 5.5, while alkaliphiles are those that like a pH above 8. So pH can have a dramatic effect on microbial growth. They can disrupt the plasma membrane. They can alter the enzymatic activity. They can also alter the membrane transport proteins. So microorganisms, they basically adapt to the pH change response by mechanism, wherein the microorganisms can maintain a neutral pH within the cytoplasmic membrane. For example, neutrophils, they can exchange potassium ions for H plus ions, and in this way, they can kind of maintain concentration of can maintain a lower concentration of hydrogen inside the H plus ions inside the cell. Next is osmolarity. So osmolarity can also be called as the water activity or the amount of water that is available for the organism. Based on these, there are three different types of environments, and isotonic environment is such in which the concentration of solutes inside the cell and outside the cell are identical. As a result, there is no inflow or outflow of water from the cell. The second condition is a hypotonic condition in which the concentration of the solutes inside the cell is higher. Solutes or salts inside the cell is higher as compared to that of the outside of the cell. As a result of which, according to the normal tendency of water, water will move from a low salt concentration to a high salt concentration. As a result, the water will enter inside the cell and it can lead to lysis of the cell by bursting. So cell wall plays here an important role in protecting the cell structure. And a third condition is known as a hypertonic solution when the concentration of ions or salts is higher outside the cell compared to the inside. So similarly, water will now try to move out of the cell in order to balance the osmolarity. And in this process, the cell is going to shrink or the protoplast is going to shrink. As a result, it is going to lose some water and hence it becomes metabolically slow. So again, there are certain categories of microorganisms that are non-halophilic, that is, they do not like or they do not tolerate or grow in high salt concentration. While there are some organisms which can tolerate salt concentration, some of the examples are Staphylococcus aureus. And then there are a third category of organisms which include halophils, which actually like growing in high salt concentration. And there are extreme halophiles that grow in very high salt concentration of more than 2 molar. So such environments are basically sea environments or saline water environments. Sorry. So the microbes have some or the other mechanosensitive channels that will allow the solutes out when they are placed in a hypotonic solution. Similarly, when they are placed in a hypertonic solution, they will try to synthesize and accumulate certain molecules such as choline, betaine, proline, glutamic acid, and other amino acids inside the cell. So they will try to balance out the ion concentrations inside and outside. So these are some of the mechanisms by which they can sustain over high or low osmotic pressure. The next factor that is affecting growth is oxygen. And based on the oxygen requirements, bacteria can be categorized into two types, aerobic and anaerobic. So aerobic are those bacteria or microbes that require oxygen for growth, while anaerobes are those which cannot grow in presence of oxygen. Broadly, there are these 5 different categories of microorganisms based on their behavior to or response to oxygen, which includes obligate aerobes. So obligate aerobes are those organisms that mandatorily require oxygen for their growth. So when you're growing cells in a liquid culture in a tube, then generally because of the diffusion of oxygen from the top, the variation in oxygen gradient is present, and hence you will see different zones where different microorganisms can grow. For example, an obligate aerobe means that it mandatorily requires oxygen for its growth. It will be only concentrated in the top zone where there is some diffusion of oxygen from the air above. Then there are strict anaerobes, which will avoid to grow in presence of oxygen. So they will be concentrated or growing towards the bottom of the tube where oxygen concentration is less. Then there are facultative anaerobes. So facultative anaerobes or facultative aerobes are those organisms which can grow in presence or absence of oxygen. So they will be present in the top as well as in the bottom layer of the medium. Then there are microaerophilic bacteria or microbes which require very less oxygen concentration for their growth. As a result, they will be not present at the very top layer, which is actually aerobic in nature, but in a zone that is below the top layer where there is some amount of oxygen diffusion. And then there are aerotolerant bacteria or aerotolerant anaerobes, which are not basically going to use oxygen for respiration. Okay, so they are anaerobes, they are going to use other compounds for respiration, but they are aerotolerant, so they are going to somehow tolerate exposure to oxygen. So they will be able to grow in the aerobic zone, but not as at a as much high concentration as that of aerobes or facultative anaerobes. So oxygen is basically very reactive, and it will generate different types of compounds which are further reactive, like superoxide, hydrogen peroxide. So microorganisms basically have some or the other mechanisms to protect themselves from the toxic oxygen products, which can damage the proteins and the DNA. So some of the enzymes include superoxide dismutase and catalase. So here is the mechanism how catalase will function. So hydrogen peroxide is one of the toxic end product that is generated because of the presence of oxygen, and enzyme catalase can break down H2O2 into water, and oxygen is released in the form of gas, which we can see over here in the form of effervescence. So this is a typical equipment that is used for culturing of anaerobes. It is known as an anaerobic workstation. So it has all the necessary conditions, such as it has a palladium catalyst so that will allow the reaction between hydrogen gas and oxygen. So oxygen is present in the environment, and that oxygen is to be removed by reaction with hydrogen, which happens in the presence of a catalyst, that is palladium. So where is this hydrogen coming from? The instrument is generally connected to hydrogen cylinders. So you put the hydrogen gas or purge the hydrogen gas inside the chamber, and in presence of the catalyst, oxygen will be removed in the form of water. So similarly, you also have this small anaerobic jar that can be used if you don't have a special equipment. And you place your petri plates in the jar, and then you have this ready-made gas pack. So you open the gas pack and you just add some water to it. That will allow release of hydrogen gas, and also there is catalyst present, so it will react with the oxygen that is present in the chamber. So you first need to add the water and seal the chamber immediately, so that the reaction will take place and oxygen will be removed in the form of water again. So in this way, anaerobic conditions can be created by anaerobic gas chambers or anaerobic workstations, which can be used for cultivation of anaerobes. Now, UV radiations, along with several other radiations such as X-rays, ionizing radiation, UV radiations are very harmful to the microorganisms. They typically cause formation of thymine dimers in the DNA, as a result of which DNA replication process is inhibited, eventually leading to cell death. So microorganisms, many of the microorganisms, they can endure the effect of UV radiation because of the presence of certain pigments such as melanin and many other pigments that are present in bacteria, which have a UV light protective action on microorganisms. So that's it about various environmental factors that affect growth of bacteria. Kindly like, share, and subscribe so that you can get a direct update of the uploaded content. Thank you.